Is it really?
Okay, so let's start with respiration. Are the numbers rolling?
Yes.
Let's go over respiration. And again, we went over photosynthesis early on, so we're going to do respiration and make sure you keep things straight, right? There's Calvin cycle, Krebs cycle, citric acid cycle, all these terms, don't get them mixed up on the exam. Dr. McGinney will try really hard to put the terms right next to each other, right? So you have choice A, citric acid cycle, B, Krebs cycle, C, Calvin cycle. Don't get mixed up, right? Okay, so let's start with respiration, and we're on, y'all should be on this page. If you didn't get a copy, I just ran out actually, sorry. Let's start with the overall goal. Again, you want to make sure you know the big picture things. What is the overall goal? Okay, so before we do that, what did photosynthesis produce? Glucose, right? Glucose contains what? Potential energy. Is glucose a long-term or a short-term energy storage? Long-term, right? So we're going to try to break down the glucose to ATP, right? ATP is short-term energy storage, and that is used to do work, right? Used to do work. Okay, so respiration then converts what? Potential energy stored in glucose to energy stored in ATP that can be used to do work, right? Okay, any questions so far? Okay, so with respiration, this is really easy to get mixed up. There's a bunch of things, so we want to make sure we understand the difference between having oxygen and not having oxygen, and its effects on respiration. So, so the first blank is respiration, cellular respiration. So happens differently between anaerobic and aerobic organisms, right? So in aerobic organisms, which means has oxygen, what do you start off with? What process? Glycolysis. Where does glycolysis occur? In the cytosol. In the cytosol of a cell. In the cytoplasm. If you have oxygen, then you're going to move on to what? The Krebs cycle, which is also known as what? Citric acid cycle. Then you move on to what? Electron transport or electron flow. Right, so that's if you have oxygen. Where does the Krebs cycle occur? In the matrix of the mitochondria, right? In the inner part of the mitochondria. Matrix of the mitochondria. Where does electron transport occur? In the inner membrane of the mitochondria. The inner membrane of the mitochondria. And that's also known as the cristae. Okay, so if you want to look at a picture of it, so this is a mitochondria. Where does glycolysis occur? Right on the outside, in the cytoplasm the products of glycolysis will move into the matrix of the mitochondria, and then from there, you're going to move to the inner membrane of the mitochondria to do electron transport, right? So be aware that the three processes are occurring in three different places, okay? So going back to photosynthesis, where did the photosynthesis occur on? Thylakoid membrane. Where did non-cyclic flow occur? Thylakoid membrane. Where did cyclic flow occur? Thylakoid membrane. Where did the Krebs cycle occur? The stroma, right? So don't get these terms mixed up, right? Sorry, where did the Calvin cycle and photosynthesis occur? In the stroma, right? Sorry, even I can mix it up. Okay. So that's if you do have oxygen, right? So if you don't have oxygen, anaerobic, no oxygen, what do you start off with? Glycolysis, which occurs in the cytoplasm or cytosol, and then you move on to fermentation. And there's two types, lactic acid or alcohol fermentation. Right, so you don't do the Krebs cycle at all. Okay. How many molecules of ATP are produced in glycolysis? A net. You produce a net of 2 ATP. How many molecules of ATP are made in the Krebs cycle? A net of 2. How many molecules of ATP are made in electron transport? 26 or 28, right? 26, 28 ATP. How many molecules of ATP are made in fermentation? 0, right? So if you do anaerobic respiration, you only make a total of 2 molecules of ATP. If you do aerobic respiration, you make way more, right? Way more. Okay, any questions? Okay, so that's the big picture thing, right? And that went in this box. So now let's look at glycolysis, right? Let's look at this glycolysis box. And within glycolysis, okay, so glycolysis is done by both anaerobic and aerobic, right? Everything starts with glycolysis. So anaerobic organisms plus aerobic organisms do glycolysis, right? And can y'all tell me the overall process? What do you start off with? Glucose, which is produced in photosynthesis, right? So glucose is converted to what? 2 molecules of pyruvate plus hydrogen ions. Okay? How much ATP is put in? 2 molecules of ATP. How many molecules of ATP are produced? 4. So what's the net gain in ATP? 2. That's the net gain of glycolysis, right? And everything that does respiration does glycolysis, okay? So with that said, what are some limiting factors of glycolysis? What could limit? Okay, so before I ask that question, so in chemistry, there is the principle, Les Chatelier's principle, right? And that was, if you add stuff to the reactant side, the reaction moves which way? So if you add reactants, the reaction moves to the products. If you add products, the reaction moves to the reactants, right? Okay. So if you add more glucose, it moves that way. Over time, you're going to have a lot of pyruvate and a lot of hydrogen ions, right? So if you have too much product, the reaction moves what direction? Backwards, right? You don't want that to happen, right? Again, if you have too much pyruvate and too much hydrogen ions, the reaction will move backwards. So your goal is to continue glycolysis. You need to make sure you're removing pyruvate and you're removing hydrogen ions, right? So glycolysis can continue. So what are some limiting factors of glycolysis? What can slow glycolysis down? Just common sense things. Okay, if you don't have enough ATP, right? If you don't have enough ATP, you can't do glycolysis. If you don't have enough glucose, you can't do glycolysis. Right? Also, if you have too much pyruvate and hydrogen ions, that's going to slow down glycolysis because the reaction will want to move the opposite direction, right? So to keep glycolysis running, you have to remove the products of glycolysis, which are pyruvate and hydrogen ions. Does that make sense? Okay. So, again, the overall purpose of glycolysis is you're breaking down glucose to produce ATP, right? So right now, you've only made 2 molecules of ATP. And let's start with organisms that don't have oxygen, right? In the earlier page, we said that if you're anaerobic, you do glycolysis. What happens after glycolysis? Fermentation, okay? So now we're going to move on to the fermentation box. And fermentation occurs for anaerobic organisms. Organisms that don't have oxygen. Okay? So, what were the products of glycolysis? Pyruvate and hydrogen ions, right? So those are going to be the products of fermentation. What are the 2 types of fermentation? Lactic acid and alcohol fermentation. Okay. So give me the reaction for lactic acid fermentation. What do you start off with? Pyruvate plus H ions produces lactic acid. Right? Alcohol fermentation. Pyruvate plus hydrogen ions produce acetaldehyde, which is ethanol, plus CO2. Right? So essentially they're the same thing. And again, what is the goal of fermentation? To remove the products, right? The goal is to remove the products of glycolysis. So glycolysis can continue. Right? They can't do the Krebs cycle. They can't do electron flow. All they can do is glycolysis, so the least they can do is keep glycolysis running, right? So fermentation doesn't produce ATP, but it allows for glycolysis to continue, so they're still getting that 2 molecules of ATP per molecule of glucose, right? So they can survive. Okay, so it's good because it keeps glycolysis running. So you're still getting the 2 ATPs. What's a bad thing, or a con? Yeah, lactic acid and alcohol are toxic to cells. Okay? So can you think of anything that does alcohol fermentation? Yeast do alcohol fermentation. Can you think of something that does lactic acid fermentation? Yeah, so our muscle cells do lactic acid fermentation when you run really hard and you run out of oxygen, right? I have one last one. So bacteria in general do lactic acid fermentation, and sometimes humans, right? Sometimes humans. Okay, any questions? Okay, so that's fermentation. Do yeast do the Krebs cycle? No. Do they do electron transport? No, right? Do bacteria that do lactic acid fermentation do the Krebs cycle? No, right? Do they do electron transport? No, right? That's all they do, right? Glycolysis and fermentation. Any questions before we move on? Okay. So now let's move on to the spinal box when oxygen is present. Okay, so when oxygen, yes. Okay, so the question is, does glycolysis produce NADH, right? So, yes, so NADPH was in photosynthesis. Yes, so NAD plus will combine with the hydrogen ions to produce NADH. Yeah, so, yes. So if you see the term NADH, that's a product of glycolysis because NAD plus will combine with hydrogen to produce a product known as NADH. Right, so don't get NADH and NADPH confused. NADPH is in photosynthesis, and a way to remember it is NADPH, like photosynthesis, right? And then NADH is just respiration. So when you do have oxygen present, you start off with glycolysis, and then you move on to what? The Krebs cycle. And then you move on to electron flow or transport. And again, where does glycolysis occur? In the cytosol. Where does the Krebs cycle occur? In the matrix of the mitochondria. Where does electron transport occur? In the inner mitochondria membrane, also known as the cristae. Right, so have these big picture ideas in your head. Okay? So if you have oxygen, you're going to move on to the next step, which is the Krebs cycle. Okay? And what happens in the Krebs cycle? Okay, so both these things occur in the mitochondria, right? So lactic acid fermentation, right? The purpose of that was to get rid of the hydrogen in the pyruvate, right? But lactic acid and ethanol were toxic. What is present in mitochondria? Oxygen, right? Oxygen from the atmosphere can combine with hydrogen ions to produce water. Right? So if you have aerobic respiration, oxygen will take up hydrogen ions to form water. And a good thing is water is not toxic to your cells, right? So that's one benefit of aerobic respiration, right? The end product water is not toxic. So that's another way you can get rid of hydrogen ions in addition to the NAD+, right? Okay, so with fermentation, the problem was you were not actually maximizing your energy production, right? Because you're not really using the pyruvate, right? You're using that to produce lactic acid, but the pyruvate isn't broken down. And that's where all your energy is, right? So a good thing is, in aerobic respiration, you're breaking down that pyruvate, right? So what happens in the Krebs cycle? pyruvate is converted into what? Acetyl-CoA and CO2. What happens to that CO2? It's released to the atmosphere, right? So again, if you're thinking in terms of the global carbon cycle, the CO2 from the Krebs cycle adds carbon dioxide to the atmosphere, right? The Calvin cycle in photosynthesis removes carbon from the atmosphere, right? So these are two opposite processes. That's released to the environment. Okay? So acetyl-CoA can now enter. Okay, so pyruvate is converted into acetyl-CoA and carbon dioxide. The carbon dioxide is released to the environment, and the acetyl-CoA can now enter the Krebs cycle. And again, so this acetyl-CoA will move from the cytoplasm into the matrix of the mitochondria where the Krebs cycle occurs. Right? So now the Krebs cycle starts with acetyl-CoA. That's converted to what? What is acetyl-CoA converted to? What's the reaction? What's the reaction? Acetyl-CoA. Okay, so what do you produce at the end? You produce citrate, right? What's in between? Oxaloacetate, right? So your acetyl-CoA, so oxaloacetate produces citrate. Okay? And so let's do some energetic calculations. So 1 ATP is produced per acetyl-CoA. Okay. 1 glucose produces how many molecules of pyruvate? 2, right? So if you look back at the reaction for glycolysis, 1 molecule of glucose produced 2 molecules of pyruvate, right? 1 molecule of pyruvate produces 1 molecule of acetyl-CoA. You get 1 ATP per acetyl-CoA. 1 glucose produces 2 pyruvates. So 1 glucose produces how many ATP molecules? Right, so 1 glucose gives you 2 pyruvates. Each pyruvate produces 1 acetyl-CoA, so 1 molecule of glucose gives you 2 molecules of ATP, right? So that's the net result of the Krebs cycle. Any questions? Okay, so this happened in the matrix of the mitochondria. So now we're ready to move on to the third part, which is electron transport. So this is also under your oxygen present box. So when oxygen is present, you go from glycolysis to the Krebs cycle to electron flow. And again, we're still in the same box. If you ran out of space, you write somewhere else. But electron flow occurs in the inner membrane of the mitochondria, which is the cristae. Okay? So what happens in electron flow? What happens? An electron is moved from molecule to molecule along the membrane. That does what? An electron is passed from molecule to molecule. This should remind you of electron flow in photosynthesis, right? This releases energy to actively transport hydrogen ions across the membrane, and that produces what? A hydrogen concentration gradient. And that hydrogen concentration gradient will power what? Chemiosmosis, right? The same in photosynthesis, chemiosmosis. And that produces ATP. And it produces a lot of ATP. How much? Yeah, about 26 or 28 molecules of ATP is produced, right? So it's moved from molecule to molecule. That energy powers active transport, which produces a concentration gradient that powers chemiosmosis to produce ATP. Okay, so that's a lot more energy, right? If you can do aerobic respiration, you do 2 plus 2 plus 26, 28, that's like 32 or something, right? That's a lot of ATP, right? So that's way more than anaerobic fermentation, anaerobic respiration that only produces 2, right? So what organisms can do aerobic respiration? Okay, let me ask you a question. Do multicellular organisms do aerobic or anaerobic? They have to do aerobic. So anytime you have a multicellular organism, whether it's a frog, a pig, or a human, something, does aerobic respiration. Single-celled organisms are usually anaerobic, but they can also do aerobic, depending on the species, okay? So multicellular can't be anaerobic. Single-cellular organisms can either be anaerobic or aerobic, okay? So make sure you know that distinction, okay? Okay, so yeast can only do anaerobic respiration, right? Yes. Okay, so the question is, at what point of the Krebs cycle is the electron release? So after you produce citrate, and there's actually a bunch of different steps. If you read the book, there's a bunch of steps. That's why he kind of skips over. Just know that citrate is produced, you produce the ATP, and then you move on. There's a lot of detail involved, and you can read. Yes, because the matrix is the inside product of the mitochondria, so that product will excite an electronic event. Okay, any questions about respiration? So what was the overall goal? What was the overall goal of respiration? Converting glucose to